My name is Sydney McAndrews. I'm about to graduate from Eastern Illinois University, which is just an hour south of here in Charleston. My project that I want to share with you is a really small stream restoration project that actually just happened like right down the road in Urbana. They did a quick habitat alteration, so they added point bars and rock structures and just made the habitat in this little area a lot better. And so today I'll just be talking about how that little stream restoration has altered the food web structure, as a case study. So first I just want to give some acknowledgments to the people that made this possible. Sea Grant, thank you guys for your funding for this project. Also the Illinois Department of Natural Resources kind of kicked it off the ground, and we had help from the EPA and the Illinois Natural History Survey. So food webs are one way that we are trying to use to help determine the success of these restoration projects. In the past, a lot of people have just looked at fish communities or macroinvertebrate communities or just the habitat itself, but it's one way to integrate all of these components into a full ecosystem level. Food webs help to describe the pattern of different trophic interactions. You can also determine the strengths of these interactions and then learn the energy pathways if they're coming from terrestrial or aquatic sources for these systems. You can also determine relationships between the actual habitat and the ecosystem processes that are happening within the habitat. So this all kind of culminates into leading to important implications in the carrying capacity of the system, which is one really important aspect of fisheries management. One way that we can actually map these food webs is through stable isotope analysis. This is starting to become kind of a big buzzword that a lot of studies are using in different areas. And so stable isotope analysis allows you to collect consumer tissue. So in this study, I'll be talking about fish and macroinvertebrate tissue that we collected. And it helps you to define the consumer's diet composition that's been assimilated over time scales. So in this, we're looking at pretty much a month time span of their diet. And so when you make these food webs using stable isotope analysis, you can create community level studies that, again, analyze that trophic complexity and the energy transfer in your system. The two most common isotopes, and the two that I'm looking at in this study, is carbon. Firstly, carbon can help you identify that basal energy source. So carbon, when you get it back from your studies, can show you either the allochthonous input or the autochthonous input. So really either the energy that's coming in from outside of the system or the energy from the aquatic source. So leaves from trees that we're finding have a very different carbon signature than periphyton or algae that's growing inside the stream. The second type of isotope that we'll be looking at is nitrogen. Nitrogen is really incredible because you can determine the trophic positioning of each of the links in your food web. So you can see who's your bottom consumer all the way to your top, top guy. So as you go up the food link, your nitrogen is enriched by about 3 to 5 per mil every time you go up trophic levels. So I have two objectives with this study. We're actually just going to look at preliminary results. I'll get my full results back hopefully by the end of next month. But with this preliminary study, I wanted to first determine the food web structure between restored and unrestored sections in the saline branch, and then also compare trophic levels among those sites. So the overall goal of this whole study was just to determine the effects of restoration on an ecosystem level dynamic within the saline branch. And I just want to share the most exciting result that we've gotten back so far, which is the positions of the fish that we've collected. So when we went out, we tried to get replicates of at least 3 to 5 replicates of fish in each section that we looked at. And the x-axis is our location. So we have 2 control sites. We have an upstream control and an adjacent control. That adjacent control is right next to our restored sites. We have 2 restored sites, an upstream restored site and a downstream restored site. From this, in this little preliminary study, we were able to get a lot of greenside darters, green sunfish, long-eared sunfish, and rock bass. And we actually found that greenside darters are on top. So you go to the next slide. It's a little surprising because these are actually really tiny fish that are beating out what you would normally consider to be the top dogs in most of these systems. So like our sunfish and our bass. This is very surprising, but it's actually not that unique. There's been 2 other studies that have found that darters have been on the top of the food chain. That's kind of showcased here. So again, another study looked at greenside darters and orange-throat darters. They found that those are both on top of the food chain in these areas. This is likely because darters are consuming the top of the food chain of macroinvertebrates. So they're eating the just Odonata, so a lot of the damselfly dragonflies, and that's enriching their nitrogen values to look like darters are on top of the food chain in these areas. So we're not done. That was very, very preliminary studies, kind of a crash course in chemistry as well. We went back out, we went to another section of stream that was restored closer to Charleston. It's actually an agricultural dominated area. So the next steps are 1, to determine if restoration is more impactful in urban or agricultural sites, and then 2, if restoration even has any effect in these areas. And so we went out, caught a lot more fish, we caught a lot more macroinvertebrates, and so right now all of our results are being analyzed at UC Davis, and so we'll be hopefully looking at that in March. But yeah, that's about it. I think, is that the last slide? Okay, great. Perfect. Yeah.